Thanks for joining me today. My name is Andy Hoisington. I am with the MVM Corps, the Military and Veterans Microbiome Consortium for Research and Education. It's a Department of Veteran Affairs program within the Rocky Mountain MIREC for suicide prevention. Basically what we work on is mental health and how that relates to microbiome, mostly human microbiome. In the last dozen years or so, I've given this talk several times, and this is a primer on human microbiome studies for non-biologists. And so I'm going to take you through some of the lessons learned, things you might want to be focused on if you're not a biologist and looking at microbiome studies. Before I get started, I am with the Department of Veteran Affairs and wanted to present this disclaimer, which we use on all of our work. The work is supported by the Department of Veteran Affairs, but doesn't necessarily mean it represents the views of the VA or the United States government. At this point, we'll shut off my video so you can see the full slides. As mentioned, I'm with the Military and Veterans Consortium for Research and Education, or MVM Corps. This group was started in 2015 by Dr. Lisa Brenner, who's the executive director, and a co-director, Dr. Christopher Lowry. MVM Corps has a mission statement listed here, which is to advance microbiome science and education to benefit military personnel, veterans, and their families. Since it began in 2015, MVM Corps now has 43 different peer-reviewed publications with dozens of active studies funded from National Institute of Health, Department of Defense, Department of Veteran Affairs, the State of Colorado, and the Alfred P. Sloan Foundation. Before I get into the details of the primer, I wanted to give you a little background here. Now, this chart is showing you the investment over a fiscal year. This is investments from the National Institute of Health in the United States. And we see in the red bars there, that's a human microbiome project. Over $200 million was invested into that endeavor. And the non-human microbiome project, over $880 million was invested. A little dated here, but it gives you an idea of some of the funding that's come across. And with that funding and other funding has exploded the literature that you can find in either the microbiome or microbiota. Here I did just a literature search a couple months ago, and it's showing you from 2000 until 2021 how many publication counts had either microbiome or microbiota in there. Now I'm going to use those words somewhat interchangeably, but if you're actually looking for the difference between those two, microbiome is a collection of genomes and all the microorganisms in the environment, while microbiota is microorganisms that are found within a specific environment. More generally, you can kind of think of microbiota as the actual bugs that we're looking at, while microbiome is the bugs plus their genes. Now, you also might have noticed that in addition to academic literature, there's a number of new articles that are non-academic. And so this was an interesting study that came out last year tracking microbiome, in this case longitudinally across the horizontal axis, and the number of stories that are published. And we see in general non-academic publications, there's been a trend where we see more publications happening, and this was actually through 2019. Now you don't have to actually go into publications and find microbiome. You can just go right to your grocery store and you'll see a large assortment of different microbiome products. In this case, this is a grocery store of probiotics where you can buy and take home with you. So let's go ahead and start from the beginning. How do we process samples? And so a sample could be a number of different things. It could be a human sample, like a fecal sample, a skin sample, or oral sample, and there's a number of different ways that we can get those samples. It could be a human associated, like we could use a filter for indoor air quality. We could collect something on HVAC filter dust. We could collect swabs. We could of furniture. So a number of different ways that we could get a sample. We then will use typically a DNA extraction kit, and there's a number of DNA extraction kits that are available to get out the DNA from that sample. Right? And there might be inhibitors, and there's some kits will do better or worse with inhibitors, things that will limit our ability to do downstream processing. We have then two options once we get to our DNA. The first option, which I'm showing you here, is to amplify DNA based on primers. So for bacteria, primers are fairly universally developed for the 16S ribosomal RNA region. That region is roughly 1600 base pair target area with 9 variable regions in itself. The primers overlap one or two of the variable regions, and selection of the primers has a large influence on the results. Not just different variable regions, but also the primers within the same variable region can have influences on the results. Overall, what we see is an amplification of generally 200 to 300 base pairs of regions, which can be read in a sequencing machine, either in forward direction, reverse direction, or some places will do a combination of the two. Here I show you one of the probably 4 or 5 main machines that are used, probably the most popular, which is out of Illumina. This is the MiSeq program or platform, but there's several others that you could use as well. Right? The other thing we could do, instead of amplifying our DNA through a primer, we can amplify the DNA directly. And so again, this is usually generally 200 to 300 piece DNA sequences, can be much smaller, even down to 50 base pairs. But what happens is we combine things together and see the overlaps. And so this is called metagenomic sequencing, sometimes also called whole genome sequencing. And I'll discuss more of the difference between these two in the next slide. Now there's other ways that we could certainly look at processing. The historical way would be culturing. Culturing would be we take the samples, we grow what we would want to find on select media. Generally, culturing was thought to contain only a small percentage of especially the bacteria that live in the environment. It's probably true for some areas, but some more researched areas like gut, actually you can have 35 to 60% of the bacteria that you can find in sequencing you can culture. Now, the nice thing about culturing is you get actually viable living and cells that can reproduce bacteria. You could also do qPCR, quantitative polymerase chain reaction. Sometimes it's also called real-time PCR. And it'll amplify specific regions like the 16S, but it used probes or primers where they read at each level of the amplification step, so you can get some ability to find out how much was in a sample, right? So you can semi-quantitatively figure out what was in the sample. You can compare it to standards, and then we can actually go back and determine how much of a certain bacteria or fungi or virus was in the sample. There's other ways, and these other ways are being investigated more thoroughly in the last decade or so. And so there's transcriptomics, the study of RNA transcripts that are produced by the genome. You study these with microarrays or reverse transcribe RNA to complementary DNA, and then you can sequence that. There's proteomics, the study of proteins. It's investigated through electrophoresis or mass spectrometry. There's metabolomics, which is a study of metabolites, including simple amino acids and related amines, lipids, sugars, nucleotides, and other intermediary metabolites. Most often you look at this through mass spectrometry. And finally, I'll mention the lipidomics, which is a study of lipids in a sample, and you can also use a version of mass spec or mass spectrometry, which is typically paired with liquid chromatography. So these are many different ways that we can look at a sample. And again, I'm going to focus here on the two most popular ones in microbiome research, which is the 16S and metagenomics. So if we look at a comparison between 16S rRNA and metagenomics here, I'll give you the basics idea of how they're different. There's many studies that are out there that will give you a lot more detail, but 16S is extensively used, has been used for a number of different studies. It targets, again, as mentioned in the last slide, specific areas in the 16S region, and so it can have primer bias. You generally get 200 to 300 base pairs, so we're looking mostly at genus level is about the lowest taxonomic level you can get to. Species level is probably a bit far. And right now, you can look at most of your sequencing technologies. You're getting between 10,000 and 20,000 sequences per sample, which is a fair bit considering a decade or so ago, I would be happy to get 1,000 to 2,000 sequences per sample. When you look at metagenomics, you get to sequence everything in a sample, so you can get information on not only the bacteria, but the fungi, the viruses, archaea, and in addition, though, as you sequence all DNA, you also sequence human DNA. And so that can be an issue. If you look at the Human Microbiome Project, it was not such an issue for fecal samples, where they removed about 1% of the samples due to being human. And there's a number of bioinformatics programs that can eliminate these sequences after sequencing. But you also look at oral and skin, and oral, those two sites had 25 to 96% of the sequences were associated with human. So you're sequencing information that you end up just kind of discarding. There's several folks that are working on how to filter that prior to sequencing, and that's an exciting technology that hopefully will be available in the next few years and widely used. You then use bioinformatics to piece together all the sequences. Again, you can be anywhere from 50 base pairs all the way up to about 300 base pairs. But what that gets you is you can get the strain level of details for some of the samples. You also can get the functional genes out of this, which is really an exciting thing for microbiome research and moving microbiome research forward. You can have anywhere from 2 million to 20 plus million sequences per sample. If remember back to I just said with 16S ribosomal RNA, we're looking at 10 to 20,000. So you get about 3 orders of magnitude larger when you sequences for metagenomics, which can be cause an issue for computational power and storage needs. So with all the great things about metagenomics, why don't we simply all do metagenomic sequencing? Well, a lot of it comes down to actually the cost here, and these are rough costs. But for about $20, you can get a sequence sampled if you already do the DNA extraction. For metagenomics, you're looking anywhere from the $300 to $400 range for samples. So it's quite a bit more expensive to do metagenomic sequencing still at this time. Hopefully those will go down. So there are a number of factors that can influence microbiome results. Here I'm just going to talk about a few of these, and these are generally the top ones that I think about here. Sampling method, shipment, storage, extractions, primer selection, sequencing, centers or platforms, and the bioinformatics pipeline when I look at a study. And so, like I said, I'm just going to talk about a few of these briefly. So for example, sampling method. Now, this has really been a benefit of the Human Microbiome Project, especially for human sampling, of where to sample, how to sample, and how often to sample. A lot of great work has been done there. So I think that one's pretty good there. Shipment, a number of people, especially for fecal swabs, will ship on dry ice. And so you have obligate aerobes that are in fecal samples that can grow, or sometimes called bloom, and so you can remove those with bioinformatics. Best though to remove those possibility of any errors there by sending on dry ice, yet most times we are having participants send in samples so they don't have dry ice, so they can either send it frozen, or you'll see preservative agents that are used for shipping, and those seem to work pretty well. We can look at, for example, DNA extraction methods. Most times, as I already mentioned, we use kits. Kits can bias either gram-negative or gram-positive. Some places now are taking the kits, the results of that, they'll sequence just the kits, the blank, and then if they get any sequences out of that, they'll remove them with the bioinformatics program. Sequencing platforms. So if you look across the different platforms, the different types of sequencers, you can get different results, more so than if you use the same sequencing technology at different sequencing centers, but we have found that you get some different results there. And then the choice of the bioinformatics pipeline has been shown by a number of people to influence the microbiome results. I often get the question of how many participants do I need for my study so I can find whatever item of interest that we're looking for. And theoretically, that would be the power calculation in statistics, and that has been done for microbiome. It actually started about a decade ago with the Human Microbiome Project. I think was the first one to come out with power calculations. There have been several improvements over time, but because of the variability of the microbiome samples, there's really not been a solid way forward determining power prior to a study to getting any information. So here I'm just going to show you the Baerdenhorst et al., which is a 2021 study, a look of over 419 different microbiome academic literature publications, and kind of we'll use that as a general framework of what people have at least done in the past. And I'll use this study several times in this presentation, so that's why I'm introducing it in a little bit more detail here now. Now, if you look at the first graph here, which is A, we see with two study groups that are looking for one outcome of interest and two a control, generally we find that it's unbalanced with about 60 samples or so per each group. And most times, and we'll come to this later, most times these are not done longitudinally. These are just at one point in time. The next panel B, we also see that overall, across the 419 different studies there also generally seems to be an imbalance with twice the amount of participants in the outcome of interest comparatively to the control group. The majority of studies that they found were generally analytical, and here in the bottom figure you can see about the sample sizes, and descriptive had an average median sample size of 33 participants, analytical was 55, predictive was 84, and treatment versus effect was 38. For the remaining part of this primer, I'm going to go through how do we analyze microbiome results. And generally I think about it in three main ways, plus I'll have an extra here. But I look at the taxonomy, alpha diversity, beta diversity. So I'm going to go through each of those in a fair amount of detail here in the next few minutes. So one way to look at our results is to compare the sequences to known bacteria or fungi through taxonomy. And you can do that through a number of different databases, curated databases. You can use OTUs or ASVs or ESVs, essentially all kind of similar results, different bioinformatics to get there, but that's just saying these sequences are associated with this taxonomy. And again, to give you the caveat here, the 16S is 200 to 300 base pairs generally, so you have to be careful when people report down to a species level, I think at least. And so here's the classification, if you haven't seen it in a while, from the middle school biology. So it starts off the domain, it goes down to species. Right, here's an example of Bifidobacterium longum, which is very commonly found in gut microbiome. Right? And so we might see something like genus, Bifidobacterium, or we might see a phylum level there. And so when we look at this, again, if you go back to the Baerdenhorst study, it looked at 419 different microbiome studies across different cohorts. You see that, or they found that in those studies, 76% of them looked at the genus level when they tried to compare across between a control and an item of interest, and 55% of them used phylum. So you will see sometimes class or order or family, but generally we see genus and then phylum. A lot of times we'll see both. And to compare it to maybe something maybe a little bit more known, here's ours, the Homo sapiens, where we rank down through from Animalia to Homo sapiens. A side note here, if you happen to be on Twitter, you might have seen some memes associated with taxonomic name changes. So there are some changes. This is at the phylum level that are coming down. I expect databases and papers will still take a few years to adjust to the new names, but they are coming. And so this is maybe one thing you might see when you look at taxonomy. This is one of our studies that looked at TBI, traumatic brain injury. And so this gives you some information here across the different samples of relative abundance. So it's set to 0 to 100% scale. The taxonomy in microbiome studies can be vast, 200, 300 or more different genera. And so one of the ways people analyze it is to look through differential abundance testing. Could be non-parametric or parametric. Those highlighted in red were most used according to the Badenhorst study of 419 different research projects. And if you are interested in learning more about differential abundance, Nearing et al. Had a recent paper in Nature Communications about 14 different differential abundance tests. Now, because we're doing multiple comparisons, most will use some kind of statistical correction for that. The linear discriminant analysis effect size, LEAF-SE, was used in 70% of microbiome studies according to the Badenhorst study. And so you'll kind of tend to see these quite often, and you can kind of recognize it because it does produce publication-ready figures. This is an example here in the right-hand side. More relevant to mental health outcomes, Nikolova et al. Reviewed gut microbiome and psychiatric disorders and came up with this table. So you see in the red, those are genus that increased. In the blue are genus that decreased. And the peach are things that were non-consistent. And the A is what was seen in multiple studies. Generally, we find that the taxa don't agree across studies. Very common finding in microbiome results. A second method to study microbiome is through alpha diversity, or that is the within sample diversity. You can look at richness, which could be the number of taxonomic groups. You can look at evenness, which measures how abundant something is distributed, or there's some that do both. There's even some that include phylogenetic differences across the tree. So let's give an example of what this might look like. So this is my undergrad at University of Michigan, their football stadium. If I wanted to look at this, I can look at this group and say how many people in this group are wearing a yellow shirt. Now I can look at where my first box is drawn there in red, and I'd see one person is wearing a yellow shirt. Right? I could look up a larger sample size and see more people are wearing yellow shirts. I could look across the whole stadium and say a lot of people are wearing yellow shirts. And that's basically the idea of rarefaction, where we have some kind of data across the sequences per sample, and in this case we have the observed OTUs that we're measuring. So we have to be careful, because if we measure wrong or with a smaller sample size, we can get skewed results. Like this, if we measure the yellow shirts in the student section, we might not have a representative sample across all of Michigan Stadium. As I mentioned, there's a couple different ways to measure alpha diversity. Here I highlighted in red, again going back to the Badenhorst study, those that were most used. Often alpha diversity is used, several alpha diversity measures are used in one study. If you look across probably the two most used bioinformatics programs, QIIME2 or Mother, QIIME2 has 32 different alpha diversity measures that you can look at. Mother has 16. This is what it might look like in a study. In the case in the human environment, the room and built environment in the squadron, we look across and look at alpha diversity measures, Shannon in this case, and you'll see a lot of times some kind of significance over it, or things that they want to highlight. You'll also see it on the right-hand side, another project we did where there's a number of measures that are being used. Generally, we find that the measures are kind of trending in the same direction. So meaning, in this case, we were looking at no TBI versus moderate to severe TBI. Historically, a higher alpha diversity has been associated with healthier phenotypes. I think in the last several years, though, people have realized that's a pretty simplistic view and doesn't hold a lot of times. Here's an example where a meta-analysis that was done where it didn't hold. You see sometimes in obese it's higher, sometimes it's lower, sometimes it's not significant. We see the same. For mental health outcomes, this one is giving you a summary of the Nikolova alpha diversity they found across multiple studies and their meta-analysis. Generally, those with negative mental health outcomes had lower alpha diversity, but not statistically significant. A third way studies might look at differences is through beta diversity, or the between sample diversity. It can be weighted, which includes information about relative abundance, or it could be unweighted. which includes no abundance information, also can include phylogenetic information. So if we go back to the stadium example, if I compared Michigan Stadium and the shirt colors that people wore there and Texas Stadium and the shirt colors people wore there, we'd see that there's some large differences. More specifically, here's the 3 most used beta diversity metrics. For no abundance, most often used was unweighted, and for abundance measurements, we see that studies use Bray-Curtis and weighted UniFrac, at least according to the Bordenhaus review, and that's what those numbers are afterwards. We often see papers that use both an unweighted and weighted UniFrac in their analysis. Looking again at the most popular used bioinformatics programs, QIIME has 25 different beta diversity metrics, and Mother has 17. We'll see these split off. Some will use unconstrained ordination like PCOA or NMSD, and others will use constrained orientation sometimes. Beta diversity can be visualized most often in an ordination plot. In this case, I'm showing you results that we had from the cadet longitudinal study. And here, this is using a non-metric diversity analysis. The horizontal axis is the first ordination, and it has the most difference, or explains the most diversity. The vertical axis then is the perpendicular line, and it shows the second most diversity. As you're looking at these plots, the closer the samples are together, the more similar they are. The further apart, the less similar. For studies like this, where we had a number of samples ranging from gut samples to skin to environmental samples, a lot of times we'll see this oversaturation impacts, also nicknamed the horseshoe effect. Sometimes you'll also see these plots with the associated statistical method, in this case per MANOVA, shown here. Here's another way to look at plots, in this case again from a study I've shown you a little bit of data from before already, moderate severe TBI versus no TBI. In this case, we show all 3 different diversity metrics, the 3 main ones at least, Bray-Curtis, unweighted, and weighted. Here you'll notice that they have percentages. That's a percent variance explained by those axes, in this case horizontal axis and vertical axis. Again, goes by the same rule. The more percent variance explained in the horizontal versus the vertical, and you almost always will find that weighted UniFrac has higher percent variance that's explained compared to unweighted and Bray-Curtis. The other way you might see this displayed, beta diversity, diversity metrics, is through an R-squared value, and that's using different techniques, but basically it's a constrained ordination method to get the R-squared values. In this case, from one of our microbiome studies for veterans, looking at the diversity associated with diet and environmental variables from both the unweighted and weighted UniFrac. So we covered taxonomy, alpha diversity, beta diversity, and those are the primary ways that you'll see studies analyzing microbiome results. But there's a number of other ways that you might see that I'm going to just kind of touch on here. First one is up there is shared sequence, and this is the sequences that are the same across the study group, often displayed in Venn diagram. Either this is just the plus or minus, does it have it or does it not? Another one could be core sequences. These are sequences that are nearly in every sample, and I mean nearly because it depends on, the percentage will depend on the author to let you know. And so sometimes you'll see core sequences at 50%, sometimes they'll be at 75%. There's not really a defined cutoff line. There's some studies looking at the gut microbiome in particular in westernized societies that reported the core microbiome, or what's the same across each individual, is about 60% of sequences. A third way you could see is cluster analysis. This is used most often in beta diversity. I already can mention some statistical modeling where you can use per MANOVA or use ANOSIM to compare these the differences across the beta diversity. But other approaches like k-means or k-nearest neighbor, which allow you to see and visualize better how samples actually cluster together in a diversity matrix. You actually could see a decade ago when I started this, or 13, 14 years ago, right, there was a little bit more of just randomly drawing circles and ovals and hoping things were close. But now I think you see a lot more scientific reasoning in the journal articles. Another one that's really become popular lately is predictive modeling or machine learning. And so a lot of studies use this, where you have independent variables from the microbiome data aimed at some kind of predictive outcomes. And the most popular prediction model is the random forest classifier. For whatever reason, you need to have a decent sample size so you can have a model or some information to train, samples to train, and then of course some to test. So that's one thing with random forest, but it's become, like I said, very popular. The other thing could be longitudinal analysis. So in the initial microbiome studies, not all, but many of them were just snapshots in time. Now we're starting to look at, people are starting to look at, I should say, not just us, but what happens over time as you perturb the microbiome. And so these consecutive samples, though, are not independent, so you have to use a little different approach. And those bioinformatics programs are certainly coming along on how to tease out what is the important issues to look for and help calculate those. I already mentioned power calculations. They're there, not robust. Most of it is post hoc right now for power calculations. And then one technique I didn't mention has been used for a number of times in 16S is this PyCrest. So what do you do if you can't afford metagenomics but you want to know functional information? This program helps identify the functional gene information from 16S. It was developed in 2013. It's probably not used as much as it used to be, although you still will see it in a number of papers. I did a quick search there, and in I think it was in about 240 or so papers that I found in 2021 in PubMed. So a number of extra things, and there's this list goes on and on, but those are kind of the main ones I'll talk about here. And so I'm going to kind of start wrapping up here, but I wanted to mention we do have a number of other quick take. I've talked about this quick take microbiome training resources coming, where we're going to talk about primers. We're going to talk about sequencing platforms and consistency across that. And these are kind of all things that over at MVM Core we've been thinking about, whether or not it's worth the costs and the effort to do metagenomics, and when do we do 16S. We're going to talk about that some, and each of these are 10 to 15 minutes, so we can just get across the information quickly. Bioinformatics pipelines, what happens with the different ones, what does research say, and other folks say about what's the difference between the different pipelines. How do we understand figures is another one I get, which is actually going to be the next one I do. So microbiome figures kind of take a life of their own, and so I'm going to help walk through how do we understand the figures that we see, the main ones that we see. Review of taxonomic methods, databases, so we'll go through some of that. Relative abundance, and what does that mean overall. We'll talk about alpha and beta, diversity, talk about in detail. Going beyond bacteria, we in MVM Core have not worked too much in viruses, but we do work with fungi, so we want to talk about that and what our results, what we found there. And then, as I already mentioned in the last slide, there's a lot of information and a lot of things going about working towards machine learning, and we've been using that. And so we want to talk about kind of we found is important, what other researchers have found important, and how to apply microbiome or machine learning in microbiome studies. So with that, I'd like to thank you for listening in. You can visit our website. We'd be happy to hear from you. Again, thanks for your time.